So it's my pleasure to now hand over to Forrest Rowher from the San Diego State University about the Holobiont Uniqueness. Looking forward.
All right, so I'm going to really jump into some old stuff and then move forward and hopefully show you something new. And of course, I'm going to try to convert everybody to being a virologist. We'll see how that goes. So remember the idea of the holobiont is that you actually have these assemblages of viruses and microbes with all the macroorganisms of the world. And so every individual macroorganism is a holobiont. We could think of it that way. And of course, the other thing that's important for this talk is the viruses, which is, and I want to remind everybody that viruses are, in fact, all of the cellular domains of life. And really, there's a whole bunch of important things about them. But for this point, for this part of it, I'm just going to focus on reminding everybody that viruses can do a normal, what we would think of as their normal life cycle, where they just go in, they infect a cell, they take the cell over, they make a whole bunch of virus particles, virions, and then they blow it open. And that's what we would call a virulent life cycle. And then there are the temperate ones, which are where the viruses go in, and then they become part of the virus for a while. And when they're in that, they form a lysogen. So that lysogen is actually the cell plus the viral genome living inside of it. And roughly speaking, all of the viruses, all the cells that are within a holobiont are actually lysogens. Okay. There's, of course, a specialty virus, which is or a special domain of viruses, which are the phage or the bacteriophage, which are just viruses that kill bacteria. And if you go out in the environment and you just start looking, what you'll find is that for about every cell out there, you'll find that there are about 10 viruses, or at least things that look like a virus under a microscope. Roughly speaking, there's something approaching 10 to the 31 viruses on the planet, which, and they have population sizes that are massive, where somewhere like 10 to the 23rd individuals. And the challenge, of course, studying viruses is that really there are a whole bunch of different morphologies, but maybe there are 20 different types of morphologies. So it's really hard to tell them apart that way. They're actually very hard to culture because, one, you have to be able to culture the cells that they're going to be raised upon. And then you have to come up with conditions that allow you to raise the virus. And then there aren't any universal markers. So there's nothing that every virus has. So you don't actually have anything like the 16S. So you have to do something like the shotgun sequencing to get there. To understand them. And so this is roughly what it would look like for us. We'll get a ship and we'll go to different parts of the world. We go underwater. We usually, most of this is done scuba diving. So we'll go down, we'll pull up, we'll collect fairly large things of water, and we'll also collect the animals. So frequently we're I tend to focus on things like corals and cnidarians. And so we'll get also samples of the animal itself so that we can look at the viruses in the animal as well as in the water around it. Then there are a bunch of techniques that you can use to get the viruses away from all the cellular material. And this is really, this is key. So this does two things for you. So one, you can actually get very clean virions. But it's almost impossible to, well, it's impossible to get cellular concentrates that don't have any viruses. So the virions can be pure. You don't really ever have that for the cells. You always have viruses there. So that's one of the main things. The other thing is that since viruses are so much smaller, it really helps to concentrate them and get them by themselves away from the cells. And then, of course, we just sequence, isolate and sequence the DNA and the RNA. And that's, of course, improving massively nowadays. And then finally, you've got all of the, what used to, we could do the sequencing stuff in about a day now, but of course it's gonna take us months to years to actually pull it apart. And that's really, you can ask who's there, what they're doing, and how many types of them are. And we put them into our bioinformatics pipelines, as well as a whole bunch of statistical and ecological modeling sorts of approaches. Now, this has been going on since this is actually the 20th year since we sequenced the first viriome, which was in 2002. And what's amazing about it is that it remains the essentially unknown. So we take all the sequences, we check it against a database, and in most environments, we would still find that about 70 to 80% of the viral sequences are unknowns. And by unknown, I mean they're really not related to anything else in the database, even using really easy or very loose criteria for calling something a known. And so this really isn't changing. So we really we're not sampling the virus very well. If you take about the ones that we have we do know something about, that we can say something about. Roughly speaking, probably these T4-like phages are the most common, followed by the lambda and the T7-like phages. All right, so if we go back and we start thinking about, well, what about those animals and plants, and really thinking about how many viruses are associated with them and so forth, we need to remember that there's viruses both inside the animals as well as associated with their skin or their digestive tracts and so forth. In the case of humans, most of the viruses are actually in the colon. So these are things that are infecting the bacteria in the colon. And there's about 10 to the 11th of them per gram of fecal matter. This is just an old study, but I really like this one. In this case, we were working with Jeff Gordon's group. And what we're doing is we're taking viriomes from their monozygotic twin study, where we've got different samples over time, and now we've got the mother and we've got the monozygotic twins. Remember, in this case, for the most part, we know that the twins tend to have bacteria a little bit more like each other, and then they're more closely related to their mother than they are to an outgroup, another family. And in this case, we're just going to sequence the viruses, and what we find is that really most of the genomic differences between individual human holobionts are encoded by these viral fractions. And this is just some of this data from this. You can do this with any holobiont out there. And what you'll find is that really the bacteria, if you're looking at what functionally they're doing, at least at this very broad scale, they're really not doing that much different, right? So bacteria have to do all cells have to do a certain number of things. When you're inside the gut, you have to set up certain ways of moving the electrons around, and you basically get very similar things when it looks at the microbiome at the cellular level. But what you see at the viral level is that you get all of these differences in the types of genes that they're carrying, and that it's extremely individualized. This is just another way of looking at it. So if I look at if we look at the same person or animal over time, what we'll find is that they tend to have the same viruses, even over pretty long periods, up to 10 years, where we have some good data on that. But then we'll find the difference between an individual and the individual next to them is extremely high. So we go from you have a certain set of viruses with you, but the diversity between different individuals is very different. Right? So what about like the other parts of the body? So what I can tell you is that nowadays we've done a lot of work, and we've really shown that like at these mucosal surfaces, we have phage that are sitting there and they're attracted to the mucus, and they have ways of just holding onto the mucus, and that protects the underlying mucosal surfaces from incoming bacteria that the phage can kill. So this is kind of an immune system that's based on the viruses that are in your gut. And the other thing that we've been looking at is, what about viruses, especially phage coming from the gut, getting inside the body? And if you measure that, what you come up with is that there are actually about 30 billion phage transcytosis events in the average human body every day. So what we've got is we've got phage coming from the gut into the blood. And this may be important for killing bacteria that are inside the animal or inside the humans. Okay. What about the viruses? So if we go into the blood, what do we find there? And this is, again, it's the same idea. You just are going to purify away all the cellular material and then look at the viruses. If you do this, you find that all humans have viruses in their blood. So we're probably producing viruses essentially all the time. So we have kind of a low scale viremia at all cases. And a lot of these things really look like they're herpes viruses, things of that nature. And when you start sequencing them, you find that we do have a whole bunch of different viruses running around, and many of them are kind of surprising. Like there are some pox viruses that are pretty common in humans. Herpes viruses are probably, they're the second most common type of viruses we find in people's blood, that everybody has a couple of them. And adenoviruses are the really interesting ones. And I just point out that coronaviruses are actually also pretty common. And I'll just end with this one, because this is the adenoviruses. So these are probably the most common viruses associated with humans. And they're also extremely common in other animals. They're just little small guys. They don't really seem to be associated with any disease of any type. And they're really understudied. So this is a great place for people to be looking at. And if you want to learn more about viruses, this is a book that we have about it. And with that, I'll stop and take questions. If there are any.
Thank you. And I see there is a question from Dominique in the panel.
Yeah, who did the wonderful drawings.
Oh, that's Ben Darby. Ben Darby's a really great artist, actually. He does both pen and ink, as well as like these really fun paintings, where really textured paintings that are fun.
Thank you.
Unfortunately, I had a technical
question.
Go ahead, Gloria.
Great talk, Forrest. It's great to see you again. So is viral diversity also being decimated by human activities? And do you think we must think of preserving viral diversity of the planet?
Yeah, so it's a good question. So I think we could in certain cases wipe out viruses that are very specific to a specific, like an animal that's lost through an extinction event. Roughly speaking, though, these viral these populations are so large that it'd be very hard for us as humans to do much to them. So the only place that you're really going to see it is where we're losing a particular animal that's a host for a virus. So I don't think that I would be too worried about most viral diversity things. And since a lot of the viruses are kind of terrible for humans, losing some of them wouldn't be so bad. Yeah.
Yeah.
And I see there's a question in Spanish or Portuguese. Sorry for my ignorance, but perhaps Karina.
There's a question, can you hear me? There's a question about the coronavirus, if it's part of the normal flora in the human blood.
Yeah, so you see coronaviruses definitely associated with humans, more in the oropharyngeal. That's the main place we see them, but we of course pick them up in blood reasonably often. Yeah, so different coronaviruses are, and they're of course widely dispersed in the animal kingdom. There are coronaviruses everywhere. So yeah, they are a normal part of the microbiome, though it's less common in the blood than it is in the oropharyngeal samples.